Genetic history of indigenous peoples of the Americas. The genetic history of indigenous peoples of the Americas, also named Amerindians or Amerinds in physical anthropology, is divided into two sharply distinct episodes. The initial peopling of the Americas during about 20,000 to 14,000 years ago, 2014 Kaya, and European contact after about 500 years ago. The former is the determinant factor for the number of genetic lineages, zygosity mutations, and founding haplotypes present in today's indigenous Amerindian populations. Most Amerindian groups are derived from two ancestral lineages which formed in Siberia prior to the last glacial maximum between about 36,000 and 25,000 years ago, East Eurasian and ancient North Eurasian. They later dispersed throughout the Americas after about 16,000 years ago. An exception are the Na Dene and Eskimo Aleut-speaking groups, which are partially derived from Siberian populations which entered the Americas at a later time. In the early 2000s, archaeogenetics was primarily based on human Y-chromosome DNA haplogroups and human mitochondrial DNA haplogroups. Autosomal DNA markers are also used, but differ from mtDNA or Y-DNA in that they overlap significantly. Analyses of genetics among Amerindian and Siberian populations have been used to argue for early isolation of founding populations on Beringia and for later, more rapid migration from Siberia through Beringia into the New World. The microsatellite diversity and distributions of the Y lineage specific to South America indicates that certain Amerindian populations have been isolated since the initial peopling of the region. The Na Dene, Inuit and Indigenous Alaskan populations exhibit haplogroup Qm242. However, they are distinct from other indigenous Amerindians with various mtDNA and at DNA mutations. This suggests that the peoples who first settled in the northern extremes of North America and Greenland derived from later migrant populations than those who penetrated farther south in the Americas. Linguists and biologists have reached a similar conclusion based on analysis of Amerindian language groups and ABO blood group system distributions. Autosomal DNA. Genetic diversity and population structure in the American landmass is also measured using autosomal DNA microsatellite markers genotyped. The Amerindian populations show a lower genetic diversity than populations from other continental regions. Observed is a decreasing genetic diversity as geographic distance from the Bering Strait occurs, as well as a decreasing genetic similarity to Siberian populations from Alaska, the genetic entry point. Also observed is evidence of a higher level of diversity and lower level of population structure in Western South America compared to Eastern South America. There is a relative lack of differentiation between Mesoamerican and Andean populations, a scenario that implies that coastal routes were easier for migrating peoples, more genetic contributors to traverse in comparison with inland routes. The overall pattern that is emerging suggests that the Americas were colonized by a small number of individuals, effective size of about 70, which grew by many orders of magnitude over 801,000 years. The data also shows that there have been genetic exchanges between Asia, the Arctic, and Greenland since the initial peopling of the Americas. Morneau-Mayer et al. 2018 have identified a basal ancestral Native American ANA lineage. This lineage formed by admixture of early East Asian and ancient North Eurasian lineages prior to the last glacial maximum, Ka. 3625 Kaya. Basal ANA diverged into an ancient Beringian AB lineage at Ka. 20 Kaya. The non-AB lineage further diverged into Northern Native American NNA and Southern Native American SNA lineages between about 17.5 and 14.6 Kaya. Most pre-Columbian lineages are derived from NNA and SNA, except for the American Arctic, where there is evidence of later after-Tenkia admixture from Paleo-Siberian lineages. In 2014, the autosomal DNA of a 12,500-plus-year-old infant from Montana was sequenced. The DNA was taken from a skeleton referred to as Anzac-1, found in close association with several Clovis artifacts. Comparisons showed strong affinities with DNA from Siberian sites and virtually ruled out that particular individual had any close affinity with European sources, the Solutrean hypothesis. The DNA also showed strong affinities with all existing Amerindian populations, which indicated that all of them derived from an ancient population that lived in or near Siberia, the Upper Paleolithic Malta population. According to an autosomal genetics study from 2012, Native Americans descend from at least 3 main migrant waves from East Asia. Most of it is traced back to a single ancestral population called First Americans. However, those who speak Inuit languages from the Arctic inherited almost half of their ancestry from a second East Asian migrant wave. And those who speak Na Dene, on the other hand, inherited a tenth of their ancestry from a third migrant wave. The initial settling of the Americas was followed by a rapid expansion southwards by the coast, with little gene flow later, especially in South America. One exception to this are the Chibcha speakers, whose ancestry comes from both North and South America. Linguistic studies have backed up genetic studies, with ancient patterns having been found between the languages spoken in Siberia and those spoken in the Americas. 2 2015 autosomal DNA genetic studies confirmed the Siberian origins of the natives of the Americas. However, an ancient signal of shared ancestry with Australasians natives of Australia, Melanesia, and the Andaman Islands was detected among the natives of the Amazon region. The migration coming out of Siberia would have happened 23,000 years ago. Paternal lineages. A central Siberian origin has been postulated for the paternal lineage of the source populations of the original migration into the Americas. Membership in haplogroups Q and C3 implies indigenous American patrilineal descent. The microsatellite diversity and distribution of a Y lineage specific to South America suggest that certain Amerindian populations became isolated after the initial colonization of their regions. The Na Dene, Inuit and Indigenous Alaskan populations exhibit haplogroup Q-Y-DNA mutations, but are distinct from other indigenous Amerindians with various mtDNA and autosomal DNA mutations. This suggests that the earliest migrants into the northern extremes of North America and Greenland derived from later migrant populations. Haplogroup Qm242 mutational name is the defining SNP of haplogroup QY DNA phylogenetic name. In Eurasia, haplogroup Q is found among indigenous Siberian populations, such as the modern Chukchi and Koryak peoples. In particular, 2 groups exhibit large concentrations of the Qm242 mutation, the Ket93, 8%, and the Selkup 66, 4% peoples. Ket are thought to be the only survivors of ancient wanderers living in Siberia. Their population size is very small. There are fewer than 1,500 Ket in Russia. 2002 The Selkup have a slightly larger population size than the Ket, with approximately 4,250 individuals. Starting the Paleo-Indians period, a migration to the Americas across the Bering Strait Beringia by a small population carrying the Qm242 mutation took place. A member of this initial population underwent a mutation, which defines its descendant population, known by the Qm3 Snp mutation. These descendants migrated all over the Americas. Haplogroup Qm3 is defined by the presence of the Rs 3894 M3 Snp, The Qm3 mutation is roughly 15,000 years old, as that is when the initial migration of Paleo-Indians into the Americas occurred. Qm3 is the predominant haplotype in the Americas at a rate of 83% in South American populations, 50% in the Na Dene populations, and in North American Eskimo Aleut populations at about 46%. With minimal back migration of Qm3 in Eurasia, the mutation likely evolved in East Beringia, or more specifically the Seward Peninsula or western Alaskan interior. The Beringia land mass began submerging, cutting off land routes. Since the discovery of Qm3, several subclades of M3 bearing populations have been discovered. An example is in South America, where some populations have a high prevalence of Snp M19, which defines subclade Qm19. M19 has been detected in 59% of Amazonian Ticuna men and in 10% of Wayuu men. Subclade M19 appears to be unique to South American indigenous peoples, arising 5,000 to 10,000 years ago. This suggests that population isolation, and perhaps even the establishment of tribal groups, began soon after migration into the South American areas. Other American subclades include QL54, Q780, Qme-H2, QSAon, and Qm346 lineages. In Canada, 2 other lineages have been found. These are Q89, 1 and Q and W tone. Haplogroup Rhone. Haplogroup Rhone Y-DNA is the second most predominant Y haplotype found among indigenous Amerindians after QY-DNA. The distribution of Rhone is believed by some to be associated with the resettlement of Eurasia following the Last Glacial Maximum, one theory that was introduced during European colonization. Rhone is very common throughout all of Eurasia except East Asia, Southeast Asia. Rhone 173 is found predominantly in North American groups like the Ojibwe 50-79%. A study of Raghavan et al. 2013 found that autosomal evidence indicates that skeletal remain of a south-central Siberian child carrying R*Y DNA Malta boy 1 is basal to modern-day Western Eurasians and genetically closely related to modern-day Amerindians, with no close affinity to East Asians. This suggests that populations related to contemporary Western Eurasians had a more northeasterly distribution 24,000 years ago than commonly thought. Sequencing of another south-central Siberian, Afantovagora II, revealed that Western Eurasian genetic signatures in modern-day Amerindians derive not only from post-Columbian admixture, as commonly thought, but also from a mixed ancestry of the first Americans. It is further theorized if Malta might be a missing link, a representative of the Asian population that admixed both Indo-Europeans and Native Americans. On ftDNA public tree, out of 626 U.S. Indigenous Americans KYS code 186, all RQ, Ronab M269, Ronia Mon 198, 1 R2 M479, and 2 most likely not tested further than Ronab M343. Haplogroup CP39. Haplogroup Qm217 is mainly found in indigenous Siberians, Mongolians, and Kazakhs. Haplogroup Qm217 is the most widespread and frequently occurring branch of the greater Y-DNA haplogroup C-Mon130. Haplogroup Cm217 descendant CP39 is most commonly found in today's Na Dene speakers, with the highest frequency found among the Athabascans at 42% and at lower frequencies in some other Native American groups. This distinct and isolated branch CP39 includes almost all the haplogroup Cm217 Y chromosomes found among all indigenous peoples of the Americas. Some researchers feel that this may indicate that the Na Dene migration occurred from the Russian Far East after the initial Paleo-Indian colonization, but prior to modern Inuit, Inupiat, and Yupik expansions. In addition to in Na Dene peoples, haplogroup CP39 C2 Bonainia is also found among other Native Americans such as Algonquian and Siouan-speaking populations. Qm217 is found among the Wayuu people of Colombia and Venezuela. Data listed here are notable indigenous peoples of the Americas by human Y-chromosome DNA haplogroups based on relevant studies. The samples are taken from individuals identified with the ethnic and linguistic designations in the first two columns. The fourth column N is the sample size studied, and the other columns give the percentage of the particular haplogroup. Maternal lineages. The common occurrence of the mtDNA haplogroups A, B, C, and D among Eastern Asian and Amerindian populations has long been recognized, along with the presence of haplogroup X. As a whole, the greatest frequency of the four Amerindian-associated haplogroups occurs in the Altai-Baikal region of southern Siberia. Some subclades of C and D closer to the Amerindian subclades occur among Mongolian, Amur, Japanese, Korean, and Ainu populations. When studying human mitochondrial DNA mtDNA haplogroups, the results indicated that indigenous Amerindian haplogroups, including haplogroup X, are part of a single founding East Asian population. It also indicates that the distribution of mtDNA haplogroups and the levels of sequence divergence among linguistically similar groups were the result of multiple preceding migrations from Bering Straits populations. All indigenous Amerindian mtDNA can be traced back to 5 haplogroups A, B, C, D, and X. More specifically, indigenous Amerindian mtDNA belongs to subhaplogroups Atwo, B2, Qneb, Qnek, Koned, Dun, and Xtua with minor groups C4, D2, and D43. This suggests that 95% of indigenous Amerindian mtDNA is descended from a minimal genetic founding female population, comprising subhaplogroups Atwo, B2, Qneb, Qnek, Koned, and Dun. The remaining 5% is composed of the Xtua, D2, C4, and D43 subhaplogroups. X is one of the 5 mtDNA haplogroups found in indigenous Amerindian peoples. Unlike the 4 main American mtDNA haplogroups A, B, C, and D, X is not at all strongly associated with East Asia. Haplogroup X genetic sequences diverged about 20,000 to 30,000 years ago to give two subgroups, Zone and Xtu. Xtu's subclade Xtu occurs only at a frequency of about 3% for the total current indigenous population of the Americas. However, Xtua is a major mtDNA subclade in North America among the Algonquian peoples. It comprises up to 25% of mtDNA types. It is also present in lower percentages to the west and south of this area among the Sioux 15%, the No-Chah-Nulth 11%, 13%, the Navajo 7%, and the Yakama 5%. Haplogroup X is more strongly present in the Near East, the Caucasus, and Mediterranean Europe. The predominant theory for subhaplogroup Xtua's appearance in North America is migration along with A, B, C, and D mtDNA groups from a source in the Altai Mountains of Central Asia. Haplotype X6 was present in the Tarahumara 1, 8% 1/53, and Huichol 20% 3/15 sequencing of the mitochondrial genome from Paleo-Eskimo remains 3,500 years old are distinct from modern Amerindians, falling within subhaplogroup D2AON, a group observed among today's Aleutian Islanders, the Aleut and Siberian Yupik populations. This suggests that the colonizers of the far north and subsequently Greenland originated from later coastal populations. and a genetic exchange in the northern extremes introduced by the fuel people Proto Inuit approximately 801,000 years ago began. These final pre-Columbian migrants introduced haplogroups Atwoa and Atwab to the existing Paleo-Eskimo populations of Canada and Greenland, culminating in the modern Inuit. A 2013 study in Nature reported that DNA found in the 24,000-year-old remains of a young boy from the archaeological Malta-Burit culture suggest that up to one-third of Indigenous Americans' ancestry can be traced back to Western Eurasians, who may have had a more northeasterly distribution 24,000 years ago than commonly thought. We estimate that 14 to 38% of Amerindian ancestry may originate through gene flow from this ancient population, the authors wrote. Professor Kelly Graff said, "Our findings are significant at two levels. First, it shows that Upper Paleolithic Siberians came from a cosmopolitan population of early modern humans that spread out of Africa to Europe and Central and South Asia. Second, Paleo-Indian skeletons like bull woman with phenotypic traits atypical of modern-day Indigenous Americans can be explained as having a direct historical connection to Upper Paleolithic Siberia. A route through Beringia is seen as more likely than the Solutrean hypothesis." An abstract in a 2012 issue of the American Journal of Physical Anthropology states that the similarities in ages and geographical distributions for Seafork and the previously analyzed X2 lineage provide support to the scenario of a dual origin for Paleo-Indians. Taking into account that Seafork is deeply rooted in the Asian portion of the mtDNA phylogeny and is indubitably of Asian origin, the finding that Seafork and Ixtua are characterized by parallel genetic histories definitively dismisses the controversial hypothesis of an Atlantic glacial entry route into North America. Another study, also focused on the mtDNA which is inherited through only the maternal line, revealed that the indigenous people of the Americas have their maternal ancestry traced back to a few founding lineages from East Asia, which would have arrived via the Bering Strait. According to this study, it is probable that the ancestors of the Native Americans would have remained for a time in the region of the Bering Strait, after which there would have been a rapid movement of settling of the Americas, taking the founding lineages to South America. According to a 2016 study focused on mtDNA lineages, a small population entered the Americas via a coastal route around 1600 Ca, following previous isolation in eastern Beringia 4-2, 4 to 9,000 years after separation from eastern Siberian populations. Following a rapid movement throughout the Americas, limited gene flow in South America resulted in a marked phylogeographic structure of populations which persisted through time. All of the ancient mitochondrial lineages detected in this study were absent from modern data sets, suggesting a high extinction rate. To investigate this further, we applied a novel principal components multiple logistic regression test to Bayesian serial coalescent simulations. The analysis supported a scenario in which European colonization caused a substantial loss of pre-Columbian lineages. Paleo-Americans. There is genetic evidence for an early wave of migration to the Americas. It is uncertain whether this Paleo-American also Paleo-Amerind, not to be confused with the term Paleo-Indian used of the early phase of Amerinds proper migration took place in the early Holocene, thus only shortly predating the main Amerind peopling of the Americas, or whether it may have reached the Americas substantially earlier, before the last glacial maximum. Genetic evidence for Paleo-Amerins consists of the presence of a parented mixture of archaic sundodont lineages to the remote populations in the South American rainforest, and in the genetics and cranial morphology of Patagonians fusions. Nemato et al. 2009 proposed migration into Beringia occurred between 40 and 30 Cal years BP, with a pre-LGM migration into the Americas followed by isolation of the northern population following closure of the ice-free corridor. A 2016 genetic study of native peoples of the Amazonian region of Brazil by Skoglund and Reich showed evidence of admixture from a separate lineage of an otherwise unknown ancient people. This ancient group appears to be related to modern-day Australasian peoples, i.e. Aboriginal Australians and Melanesians. This ghost population was found in speakers of Tupian languages. They provisionally named this ancient group Population Y after Epicura, which means ancestor in the Tupi language family. Archaeological evidence for pre-LGM human presence in the Americas was first presented in the 1970s. Notably, the Luzia woman skull found in Brazil and the Monte Verde site in Chile, both discovered in 1975. Other notable sites of early human inhabitation found in North America include Paisley Caves, Oregon, and Bluefish Caves, Canada. Genetic analyses of HLA-I and HLA-Roman II genes as well as HLA-A-B and DR bone gene frequencies links the Ainu people in northern Japan and southeastern Russia to some indigenous peoples of the Americas, especially to populations on the Pacific Northwest coast such as Tlingit. The scientists suggest that the main ancestor of the Ainu and of some Native American groups can be traced back to Paleolithic groups in southern Siberia. The same lineages are also found among some Central Asians. Old World genetic admixture. Substantial racial admixture has taken place during and since the European colonization of the Americas. South and Central America. In Latin America in particular, significant racial admixture took place between the indigenous Amerindian population, the European-descended colonial population, and the sub-Saharan African populations imported as slaves. From about 1700, a Latin American terminology developed to refer to the various combinations of mixed racial descent produced by this. Many individuals who self-identify as one race exhibit genetic evidence of a multiracial ancestry. The European conquest of South and Central America, beginning in the late 15th century, was initially executed by male soldiers and sailors from the Iberian Peninsula, Spain, and Portugal. The new soldier settlers fathered children with Amerindian women and later with African slaves. These mixed-race children were generally identified by the Spanish colonist and Portuguese colonist as castas. North America. The North American fur trade during the 16th century brought many more European men from France, Ireland, and Great Britain, who took North Amerindian women as wives. Their children became known as métis or boys brules by the French colonists and mixed bloods, half-breeds or country born by the English colonists and Scottish colonists. Native Americans in the United States are more likely than any other racial group to practice racial exogamy, resulting in an ever-declining proportion of indigenous ancestry among those who claim a Native American identity. In the United States 2010 census, nearly 3 million people indicated that their race was Native American, including Alaska Native. This is based on self-identification, and there are no formal defining criteria for this designation. Especially numerous was the self-identification of Cherokee ethnic origin, a phenomenon dubbed the Cherokee syndrome, where some Americans believe they have a long-lost Cherokee ancestor without being able to identify that person in their family tree. The context is the cultivation of an opportunistic ethnic identity related to the perceived prestige associated with Native American ancestry. Native American identity in the eastern United States is mostly detached from genetic descent and especially embraced by people of predominantly European ancestry. Some tribes have adopted criteria of racial preservation, usually through a certificate of degree of Indian blood, and practiced disenrollment of tribal members unable to provide proof of Native American ancestry. This topic has become a contentious issue in Native American reservation politics. Ancient Beringians. Recent archaeological findings in Alaska have shed light on the existence of a previously unknown Native American population that has been academically named Ancient Beringians. Although it is popularly agreed among archaeologists that early settlers had crossed into Alaska from Russia through the Bering Strait land bridge, the issue of whether or not there was one founding group or several waves of migration is a controversial and prevalent debate among academics in the field today. In 2018, the sequenced DNA of a native girl, whose remains were found at the Sun River Archaeological Site in Alaska in 2013, proved not to match the two recognized branches of Native Americans and instead belonged to the early population of ancient Beringians. This breakthrough is said to be the first direct genomic evidence that there was potentially only one wave of migration in the Americas that occurred, with genetic branching and division transpiring after the fact. The migration wave is estimated to have emerged about 20,000 years ago. The ancient Beringians are said to be a common ancestral group among contemporary Native American populations today, which differs in results collected from previous research that suggests that modern populations are descendants of either northern and southern branches. Experts were also able to use wider genetic evidence to establish that the split between the northern and southern American branches of civilization from the ancient Beringians in Alaska only occurred about 17,000 and 14,000 years, further challenging the concept of multiple migration waves occurring during the very first stages of settlement. Blood groups. Prior to the 1952 confirmation of DNA as the hereditary material by Alfred Hershey and Martha Chase, scientists used blood proteins to study human genetic variation. The ABO blood group system is widely credited to have been discovered by the Austrian Karl Landsteiner, who found 3 different blood types in 1900. Blood groups are inherited from both parents. The ABO blood type is controlled by a single gene, the ABO gene, with 3 alleles, I, Ia, and Ib. Research by Ludwig and Hanke Hirschfeld during World War I found that the frequencies of blood groups A, B, O differed greatly from region to region. The O blood type, usually resulting from the absence of both A and B alleles, is very common around the world, with a rate of 63% in all human populations. Type O is the primary blood type among the indigenous populations of the Americas, in particular within Central and South America populations, with a frequency of nearly 100%. In indigenous North American populations, the frequency of type O ranges from 16% to 82%. This suggests, again, that the initial Amerindians evolved from an isolated population with a minimal number of individuals. The standard explanation for such a high population of Native Americans with blood type O comes from the idea of genetic drift, in which the small nature of Native American populations meant the almost complete absence of any other blood gene being passed down through generations. Other related explanations include the bottleneck explanation, which states that there were high frequencies of blood type and B among Native Americans, but severe population decline during the 1500s and 1600s caused by the introduction of disease from Europe resulted in the massive death toll of those with blood types A and B. Coincidentally, a large amount of the survivors were type O. European diseases and genetic modification. A team led by Ripan Malhai, an anthropologist at the University of Illinois in Urbana, conducted a study where they used a scientific technique known as whole exome sequencing to test immune-related gene variants within Native Americans. Through analyzing ancient and modern native DNA, it was found that HLA-DQ-AON, a variant gene that codes for protein in charge of differentiating between healthy cells from invading viruses and bacteria, were present in nearly 100% of ancient remains, but only 36% in modern Native Americans. These findings suggest that European-born epidemics such as smallpox altered the disease landscape of the Americas, leaving survivors of these outbreaks less likely to carry variants like HLA-DQ-AON. This made them less able to cope with new diseases. The change in genetic makeup is measured by scientists to have occurred around 175 years ago,